Hi everyone, this is Muthu Kumar, Associate Professor of Pharmaceutics Department, belongs to SNS College of Pharmacy and Health Sciences. Today we are going to discuss about the indicators used in the sterility testing and pharmaceuticals. So what are the indicators we are going to use for the sterility testing and pharmaceuticals? Not only pharmaceuticals, nowadays nutraceuticals departments also we are using the indicators to evaluate the sterility testing methods. So what is the purpose of indicators used in sterility testing of pharmaceuticals? So the first one, it ensures the microbial contamination free pharmaceutical product as well as the nutraceutical product. Not only in pharma, nowadays in almost all categories, they are using the sterility indicators to evaluate the microbial contamination and efficiency of the sterilization equipments, etc. Next, role of biological and chemical indicators in validating sterility testing. What are the role of biological indicators? Nothing but a microorganism, and chemical indicators nothing but a liquid and gases chemicals comes under the category of the indicator side. So what are the role of the biological as well as the chemical indicators to validate the sterility testing in the pharmaceutical units? Next, used in clean rooms, means there are so many working rooms in the pharmaceutical sectors. Next, to evaluate the sterilization cycles, how it evaluates the sterilization cycle in the continuous production as well as the batch production, and SAL programs. SAL programs is nothing but a sterilization assurance level, how it used to identify the SAL programs. Next, overview of the sterility testing. What is the purpose of the sterility testing first? First purpose is nothing but to confirm the complete free or absence of the viable microorganisms in sterile drug products, especially ophthalmics, injectables, LVP, SVP, whatever may be. It should be completely free from the viable microorganisms. That is the first aim. Next, record for the pharmacopoeia. We know the pharmacopoeia. What are the important pharmacopoeia like USP, European Pharmacopoeia, in India, of course, Indian Pharmacopoeia. So what are the requirements they mentioned under the Pharmacopoeia? It has to meet the proper requirements. Next, the purpose of the sterility testing is it performed on parenterals, ophthalmics, implants, vaccines and sterile devices in the industry side. It has to be completely free from pathogenic microorganisms. Next, what are the methods involved in the sterility testing? So first is the membrane filtration method. It's only applicable for aqueous and oily pharmaceutical products. Next, direct inoculation into culture media. There are the special culture media for each and every category. So the suspension of the product, pharmaceutical product, it will be directly inoculated on the media, then it will go for incubation and identification. Next, importance of the indicators. So it verify sterility process. So how the sterility process is going on, whether it is in efficient or inefficient category. Next, the same like it assess the environment and test reliability in the pharmaceutical industry. Next, indicator side. The first indicator is nothing but a biological indicators. So what are the biological indicators? So biological indicators is nothing but it is a standardized preparation of the resistant bacterial spores or the microbes which has the highly resistant spore bearing category. They pick such type of microbes, then it will use for validate the sterilization systems. So we know when compared to the other categories, spore bearing microbes are highly resistant towards the temperature and pressure. That is the reason why we pick those type of microbes as a biological indicators. So what are the common Biological indicators. Geobacillus stereothermophilus, it's mean for steam sterilization, and Bacillus atrophius, it's for gaseous sterilization and dry heat sterilization. So what are the key properties? It's a D value. So known D value, it's nothing but the time take to reduce the population or the microbial load reduce 90%. So what are the known population load? So it has to be in the range of 10 to the power of 5 to 8 spores per million. Next, high resistance ensures cycle effectiveness. So what are the key properties here? First, known D value, known population load, and the final one is the high resistance ensures cycle effectiveness. So what are the high resistance means? These microbes, they are slightly vary when compared to the other microbes. Because of the spore bearing properties, they have a high resistance towards the cycles, means pressure withstanding, temperature withstanding throughout the process. They are the key properties of the biological indicators. So what are the application? These biological indicators applied in the autoclave cycle validation and deep hydrogenation tunnels, especially in the hot air sterilization and gas sterilization evaluation. Next, it load release in protein production. So each and every category, whether it is a continuous or the batch production, so it has to be completely evaluated and documented. For that purpose, these type of biological indicators extensively used in the pharmaceutical industries. Next, chemical indicators. What are the chemical indicators? So chemical indicators is nothing but the chemical impregnated or embedded in a particular sensor. So once it receives or undergoes the thorough sterility testing, it will be change its nature, thereby it gives the result whether the sterilization is properly evaluated or not. So types, what are the types? So as per the ISO, there are 1 to 6 types of chemical indicators. So class 1 is nothing but a process indicator. Class 2 is nothing but a OV-DIC indicators, especially to identify the steam penetration. Class 3 to class 6, it's a parametric indicators, exclusively for temperature, time, and ethylene oxide gases gases concentration identification. What are the functions of the chemical indicators? So it provide a quick and visual confirmation of the exposure. So there is no need to go for the membrane filtration, direct inoculation assessment. So immediate response. Once the sterilization evaluation over, immediately we get the result. It doesn't replace the biological indicators. Biological indicators area is a different category, and it's used for batch manufacturing and packing validation system in pharmaceutical sector. Next, what are the environmental and process indicators? So environmental monitoring indicators, it's nothing but air sampling, viable particular counters. Among the all, they are the two very important. Air sampling is nothing but an active and passive ways it has to be performed. So we take the air sampling, thereby we are going to identify the environment is free from pathogenic microbes or not. Next, viable particle counters. So there is a special particle counters. It is going to be assess the viable microbes presence in the environmental or not. Next, media growth promotion test. So it confirms the culture media and support microbial growth. Thereby it ensures there is no media failure during the sterility testing. That is the main advantage of the media growth promotion test. Next, positive and negative controls for the environmental process and process indicators identification. So positive control, it ensures detection capability. So Positive means we know the presence of the particular things. So if there is a positive control, it ensures detection capability. Next, negative control means it ensures no contamination during handling procedures. So negative control, it's nothing but a indirect identification of the sterilization process. There is a no contamination during the handling or the entire process, that will be considered as the negative control. So conclusion about the indicators. So indicators collectively ensure sterility assurance levels. Thereby, it gives an indirect report that a particular process is effectively undergoing the sterilization, and it gives the product completely microbe-free category. Next, process validation. Process validation, it evaluates the equipment. So throughout the process, what are the equipments extensively used for the pharmaceutical preparation has to be evaluated, and it has to be noted under the regulatory complaints in the category of the microbial free situation or aseptic free environment. So if there is any asepsis inside the microbes preparation area or the pharmaceutical packaging area or the production area, so the entire efficiency what they are going to put for the production, it's completely waste. So in order to assure or ensure the product it efficiency or makes the product microbial free category, the process has to be extensively validated, and it should be adherence towards the particular country's regulatory compliance.